Let's talk about regulation of metabolic pathways, things like glycolysis and gluconeogenesis, which remember are happening in the context of a broader metabolism, where not only does that glucose that either enters your cells or is released from glycogen, not only can it go through glycolysis, but it can go through the pentose phosphate pathway to make NADPH. It can go and get hooked up into glycogen, all these different options for glucose. We're going to focus today on just glycolysis and gluconeogenesis. So glycolysis, the breakdown of glucose to give you pyruvate, and gluconeogenesis, the making of glucose from pyruvate or other non-carb sources. Just by focusing on these two pathways, we're going to encounter pretty much every common theme of metabolic regulation that we're going to talk about in this class and that you'll see in metabolism. One, we're going to regulate those far from equilibrium steps. So those steps with a really large delta G, those steps that are hard to go back from. Those steps, for example, where we actually have to take a different enzyme and we go for glycolysis versus going for gluconeogenesis, because it's going to be so energetically oomphy in one direction, we're going to not just kind of regulate the direction by changing concentrations, but we're going to regulate the direction by regulating the enzymes themselves. Those steps are often going to serve as kind of committed steps. So remember when we talked about glycolysis, we said that the committed step was going to be this phosphofructokinase 1 step. And you can see here it's going to have a lot of regulation. So we'll see how it can be stimulated by F26BP and AMP and ADP, and it can be inhibited by ATP and citrate. And so we'll look and see why these different allosteric modulators, we say, these small molecules that can bind to and alter the function of the enzyme, why they make sense and how they do what they do. But we call this step the committed step because if you look before it, this glucose 6-phosphate, although this top step, this hexokinase step, we'll see how it's going to be highly regulated because it's kind of trapping the glucose in the cell and you are investing energy. So that is, say, one of those points where we do have one of those metabolically irreversible steps. However, when we trap the glucose, now the glucose has multiple fates. And so it can go in the pentose phosphate pathway, which remember will give us the NADPH we can use for anabolism, things like making lipids, as well as for combating oxidative stress by regenerating glutathione and things. It's also really great if you want to make nucleotides, so you want to make some DNA or RNA, so rapidly dividing cells are often going to want to use the pentose phosphate pathway. We could also take that glucose 6-phosphate and hook it up into glycogen. We can interconvert it with phosphoglucose isomerase to give us fructose 6-phosphate. As indicated by the double-sided arrows, this is going to be pretty easy to reverse, and so this is going to operate in both glycolysis and gluconeogenesis. However, once you get to fructose 6-phosphate, well, here you're actually going to have it so that if you were to go the phosphofructokinase route, it's harder to go back to another pathway, and so we call this the committed step of glycolysis. Note too that because this phosphoglucose isomerase step is fairly neutral, it's kind of easy to go back and forth. Depending on the concentrations of these intermediates, we can determine which way the pathway is going to go based on cellular needs. So that's one way we can regulate is just by kind of regulating supply and demand, regulating the concentrations. One way that we can regulate things as well is going to be using hormones. So we'll talk about, remember how we have insulin, which is kind of going to signal to the body, hey, let some glucose in. We'll see how the insulin is actually going to tell your liver to let glucose in. And glucagon, all the glucose is gone, that's going to tell your liver to actually ship out glucose. And so we'll look and see how that's going to allow the liver to ship out glucose. So those are some of the common themes. We'll get a little more into depth about types of allosteric regulation. So we can have the negative, which is kind of where something farther down in the pathway is going to signal to the upstream, like, hey, you need to decrease our supply because we don't have enough demand down here. So going back to our example of kind of the umbrella factory where there's a storm coming outside and you want to get to the message inside to the umbrella makers to start making umbrellas, but maybe they overshoot it, they make too many umbrellas. You want to have the umbrellas then go and inhibit the making of the umbrellas. That would be an example of negative allosteric modulation. Alternatively, if you have a lot of demand, you might have kind of some of the upstream intermediates be feed forward, being saying like, okay, there's a lot of stuff coming down the line, you better get prepared. So maybe we're making a lot of the individual pieces of the umbrellas. We have a lot of the handles, we have a lot of the hooks, and we need to tell the people that put all the pieces together, hey, we've got a lot of pieces coming to you, so get ready. So that would be an example of a positive allosteric modulation. We'll look and see how we can use diagrams. So we can see like a red line with a little line at the end, or an X would be a negative allosteric modulation or negative regulation of some sort, whereas a green line would be a positive regulation of some sort. We'll see that you can kind of integrate signals from different pathways to piece things together. So we'll look and I'll point out specific examples of each of these things in the case of glycolysis and gluconeogenesis. We're going to talk about how these are kind of going to be reciprocally regulated, because you don't want to have a fetal cycle where you're breaking down glucose at the same time as you're making it, especially because you're kind of going to want to make glucose or break down glucose at different times. You might want to do glycolysis when, well, there's a lot of glucose, but you don't have enough energy. And if you're the liver, you might want to do glucogenesis when you have plenty of energy, but not much glucose. So you need to make some glucose to send out to other cells. This is an example of kind of compartmentalization. I mentioned the liver because that's where this gluconeogenesis is actually going to take place. And one of the ways that we can regulate metabolic pathways is by compartmentalization and specialization. We can specialize so that only certain cells or certain tissue types are able to actually carry out various processes, because only they will have the enzymes responsible to basically carry out some of the reactions required. Additionally, even if some tissues have those enzymes, we can have different versions of the enzymes in different tissues that are then able to be regulated separately and have different kinetic properties. So we'll take a look at this in our example. Let's start by just thinking logically about when we would want to do which. In terms of glycolysis, so remember glycolysis, this is going to be when we're breaking down glucose, and often we're doing that to get energy. And so if we have a lot of glucose but not enough energy, then we want to do glycolysis. And how would we know this? Well, if we have low ATP and high ADP or high AMP. So remember that our ATP to AMP ratio is going to be a more sensitive indicator because of energy status, because we can kind of take two ADPs and combine them to give us an ATP and an AMP. And the amount of AMP is going to be lower than the amount of ADP. And so small changes in it can have a bigger effect. And so that ATP to AMP ratio is going to be a good indicator of cellular energy status. And that's going to help tell us that we should do glycolysis. Remember that glycolysis, you need NAD plus and you make NADH. If you have a lot of NAD plus and low NADH, well, then you're going to want to do glycolysis. What about gluconeogenesis? Here's the opposite of things. If you have a lot of energy, but you don't have much glucose, if you have high, so that would be like high ATP and low ADP and AMP ratios, or if you have low NAD plus and high NADH ratios. We'll see that both ADP and AMP are going to be allosteric regulators. So they're going to be able to bind to enzymes and alter their activity. In your liver, you're also want to do gluconeogenesis when you have like glucagon signaling telling you that there's not much glucose in the bloodstream, and so you need to make glucose for the bloodstream in order to let it out. This gets us to the idea of kind of tissue-specific metabolism and having tissues specialize in doing different things, and having tissues have different versions of enzymes or like isozymes that'll allow them to carry out their specific functions. A couple of key tissues that come into play in metabolism are your liver and your muscles. Your liver and your muscles serve as good kind of comparison points because both of them are going to store glycogen, so they're going to store glucose, and thus have the potential to be breaking down the glucose and doing glycolysis from those stores in addition to kind of glucose that they just take in from the bloodstream. However, your liver and muscles have very different priorities. Your liver, it has the role of regulating blood sugar throughout the body. It's got to be able to respond to low blood sugar and to let glucose out into the bloodstream. And it's got to be able to, if they're high blood sugar, take that sugar in. Your muscles, they're also going to take glucose in, but they're going to store it for themselves in glycogen. And then when they need energy, they'll break down that glycogen in order to get glucose that they'll use for themselves. If we want to regulate how much glucose is actually in those cells at any time, we need to regulate how much glucose gets into the cells as well as how much glucose gets trapped into the cells. This leads us to thinking about the regulation of glucose import and the first step, hexokinase, where we're going to actually phosphorylate the glucose at that 6th position to give us our glucose 6-phosphate, trap it in the cells, and then they can have their different fates. The regulation of glucose metabolism starts at the import. It starts with actually the glucose transport. There are different isoforms or versions of the glucose transporter that are produced in different tissues. And these reflect the different roles of those tissues. For example, we talked about how the liver needs to be able to take in excess glucose when there's high glucose in the bloodstream. This will prevent your blood sugar from building up too high. If you look here, you can see that GLUT2, this glucose transporter, has a high KT. A KT, this is the equilibrium constant for the transport of glucose into the cells. And you can see that it has a high KT. This high KT indicates that it has a pretty low affinity, which means that it's not going to hog all of the glucose, and it means that it's going to be hard to saturate it, so that if there are high levels, it's going to keep taking in that glucose, while other tissues, you're kind of working at capacity. Speaking of your other tissues, think about your brain. It's really important that your brain has glucose because, well, your brain's really important and it can't use other fuel sources very efficiently. So your brain is very reliant on glucose, and you want your brain to be able to take glucose even if there's not very much in the bloodstream. So you'll see that it has a low KT, indicating a high affinity. So even if there's not very much glucose in the bloodstream, your brain's going to take it in. This low KT, however, also means that it's going to get saturated really quickly. And so if the concentration is higher, it's not just going to keep taking it in and taking it in and taking it in because, well, even if it takes it in, it'll then let it out because it is such a low KT, it's going to get saturated quickly. And if it gets saturated, that's kind of the maximum level. So it's basically always taking its maximum level, but that maximum level is still leaving plenty in the bloodstream for other tissues to have. Think about your brain. Your brain's going to need a lot of sugar. Your brain's going to need a lot of energy. And so it can't store glucose. It can't even use like fatty acids and stuff. And so what's going to happen is your brain is going to really, really rely on glucose. It's going to need to have a really high affinity transporter. So no matter how much glucose is in the bloodstream, it's going to take it. It's going to take it. And so we have this low KT. Most of your tissues are going to express this GLUT1. This GLUT1 is going to be basically, when we say it says like basal glucose uptake, so that's kind of like your baseline, your just kind of all the time uptake. And so you can see you have a kind of medium affinity, and so you are going to be taking in the glucose, getting what you need. And finally, the one that's more exciting is going to be this GLUT4. This GLUT4, this is going to be found in your muscle, in your fat, in your heart, and its activity is going to be increased by insulin. So insulin, remember, was saying that the blood sugar is high, so let some glucose in. And what you're going to do is, well, your liver would want to take it in to store it as glycogen, and your muscles will want to take it in to store it as glycogen. Now, your liver is going to be able to take in the glucose when there's a lot of glucose just because it has that high KT of this GLUT2 transporter. However, your muscles, they're going to be their normal transporter. They're like GLUT1 is going to be saturated. It's going to be at capacity and it's not going to take more in. However, GLUT4 is going to have its activity increased by insulin. And so now we see an example of our hormonal regulation of metabolic pathways. Insulin is going to signal for the bringing of the GLUT4 transporter to the membrane. So you kind of have these like little membrane pouches inside of the cells where there's the GLUT4 transporter pre-made. This way, your cells don't have to go through the whole process of making it when they need to respond really quickly to insulin. Instead, they just have to have some insulin signaling pathway lead to the bringing of the GLUT4 transporters into the surface of the cells, and that's going to allow the sugar to be let in. Even though the transporter itself is not directly regulated by insulin in the case of your liver, The making of it can be, so you get transcriptional regulation from the insulin signaling that is going to regulate the levels of the transporter that are made and the levels of the phosphatases and stuff, so that you're regulating things still hormonally in the case of your liver, even though you don't have this kind of insulin responsive transporter. It's actually from a paper where people went and they measured these things, and you could see that as glucose is ingested, insulin levels spike and glucagon levels go down. When the insulin levels spike, the plasma glucose, so the amount of glucose in your bloodstream, goes down. These transporters are doing passive transport, so not only can they let glucose in, but they can let glucose out. Because this KT is really high for this GLUT2, it's going to make it so that the liver and your intestine and kidneys and pancreas, they're not going to be hogging all of the glucose. But they are able to take in glucose when the other tissues won't be. But in order to trap it, they're going to actually have to phosphorylate it. So this gets us to regulation of our hexokinase. Hexokinase, remember, that is going to be the enzyme that's going to phosphorylate glucose at the 6th position to give us our glucose 6-phosphate. This has the role of trapping the glucose inside of the cell, investing some energy, making it so that glucose 6-phosphate isn't going to flow out, and that you'll have energy for doing things later on in the pathway. There are 4 different hexokinase isozymes, so versions of these enzymes. So again, going back to kind of tissue specialization, and these are regulated differently and have different kinetic properties. Our main ones, hexokinase 1 and 2, as well as hexokinase 3, they're all going to be inhibited by their product, glucose 6-phosphate. Glucose 6-phosphate is the product, it's inhibiting the enzyme. This is an example of negative feedback inhibition. Hexokinase 4, however, and this is also called glucokinase, this is found in primarily, we talk about it in your liver, but it's also in your pancreas and small intestine and hypothalamus. It is not inhibited by glucose 6-phosphate. So it does not have that level of negative feedback inhibition, and it also has different kinetic properties. Let's think about why it makes sense for it not to have this negative feedback inhibition from glucose 6-phosphate. Remember that the liver is responsible for kind of making sure the blood sugar doesn't get too high by taking in excess glucose. If the liver hexokinase was inhibited by the glucose 6-phosphate, it would take in the glucose, it would trap it, and then it would stop trapping the glucose. It would stop trapping the glucose because the cells would be like, hey, we've got enough, we've got enough, we've got enough. And so even though there's a bunch in the bloodstream, we have too much in here, so stop taking it in. But the liver's job is to be the one that takes it in. And so we need to prevent it from getting inhibited by that glucose 6-phosphate. And so it's going to have a different isozyme, and it's not going to be inhibited by the glucose 6-phosphate. However, we need to make sure that the liver's not getting too hoggy. We need to make sure that the liver's not just going to take in whatever glucose comes its way, and we need to make sure that the liver is able to release the glucose and not just keep it all for itself. How are we going to do this? How we're going to do this is by having different kinetic properties of the enzyme. First, let's orient ourselves to what we're looking at. The glucose concentration is on our x-axis, and the initial velocity is on our y-axis. So we're looking at one of those kind of Michaelis-Menten plots. Thinking back to our Michaelis-Menten kinetics, we have the values Km and Kcat. Km was the one that was going to tell us kind of about affinity for a substrate, with the lower Km indicating a higher affinity. And the Kcat, that was going to correspond to kind of the turnover rate, so how quickly it could act to actually turn the substrate into product. If we look at this, what we can see is that hexokinase 1, so kind of that version that's in most of your tissues, is going to have a lower Km than your hexokinase 4, meaning it has a higher affinity. It's saturated more easily. The hexokinase 1, this is going to, however, saturate at a lower level than you have your hexokinase 4, your glucokinase. This makes sense. Why does it make sense? Well, if you think about your enzyme kinetics, you will have that point, you have that kind of like steady state where you have things going linear and then you kind of saturate out. In the case of our hexokinase, it's this linear region in the region of concentrations that you'll find in your body. So around this 5 millimolar is kind of your normal fasting blood glucose levels. And in this range, your hexokinase 1, so the version that most of your cells have, is kind of plateaued out already. So it's already working at its maximum capacity, and it's not going to be very representative of what's going on outside the cell. However, if we think about our hexokinase 4, our glucokinase, now we're in the linear range. This means that changes in the concentration of glucose in the blood will change the activity of this enzyme. If we think about hexokinase 1, well, at this, changes in the level of the glucose are not going to impact the activity of the enzyme because the enzyme is already working at its maximal activity. It's already at its Vmax. But you've got to get to crazy high levels to get to the Vmax of your hexokinase 4. And this means that it's going to be able to respond to changes in blood sugar by changing its activity. It's going to therefore serve as a glucose sensor. Importantly, too, because it's got a lower affinity, it's not going to hog all the glucose. So it's going to allow those other cells to take in what they need. Those other cells will take in what they need. Then they'll get inhibited by the glucose 6-phosphate. They'll be working at their maximum, so they can't trap anymore. And so what they can't trap is going to get let back out to the cells, remember. The liver is going to have a transporter that's going to allow it to keep taking it in. The liver is going to take it in. It's going to respond and phosphorylate it. However, it's not going to hog all of it because it has the lower affinity. And if conditions go the other way, where you have a lower blood sugar, well, now what's going to happen is that the liver is going to actually be able to let out the glucose because it doesn't have the super high affinity where every time it dephosphorylates the glucose, it's actually going to phosphorylate it again and trap it. And so the dephosphorylation is actually going to be happening kind of controlled in a manner that's going to keep it protected from getting phosphorylated on its way out of the cells. So it's actually found in an intracellular compartment, so your endoplasmic reticulum, in order to kind of get chaperoned so that it doesn't just get trapped back. Because you want to release it, and if you need to release it, you want to prevent yourself from just phosphorylating it back. So having this lower affinity of your hexokinase is going to help with that, as well as having a higher KT of your transporter so that you are able to let out this. So you're able to take things in, you're able to let things out, and that's what it's all about. Glucose import and export and all that. Yeah, okay. Enough of that. Let's move on. Our next step that we need to regulate is this big one, that fructose 6-phosphate to fructose 1,6-bisphosphate. So remember, in glycolysis, that's carried out by phosphofructokinase 1. And then to go the reverse way, we're going to use fructose 1,6-bisphosphatase 1. And these are going to be highly regulated and reciprocally regulated. So why are they so crazy highly regulated? Because, as I mentioned, this was the committed step of glycolysis. So basically, a lot of things we need, glucose can go a lot of different directions once you trap it inside of the cells. One of the main other ways it could go, other than glycolysis, is the pentose phosphate pathway. This is going to be used to make NADPH, DNA, RNA, fatty acids, steroids, etc. So there's all these sorts of other things we can do with glucose rather than just break it down for energy. If we want to use glucose for any of those things, we're going to want to phosphorylate it. So that wouldn't be the main commit. That's not a committed step to glycolysis. Instead, after we do this isomerase step, the next step is the committed step where we're saying, okay, now let's take this fructose 6-phosphate and change it to fructose 1,6-bisphosphate. In order to kind of regulate what direction things will go, one of the main regulators of this is going to be NADPH. One of the things, remember, of the pentose phosphate pathway is it's actually making NADPH. And so if you make a lot of NADPH, well, now we get this negative feedback regulation, which says, okay, we've done enough of this pentose phosphate stuff. But even once we make that decision, we still need to decide, okay, well, are we going to go towards glycolysis? Are we going to go towards gluconeogenesis? Do we want to upregulate PFK1 or do we want to upregulate FBPase1? And so PFK1 would basically take us towards glycolysis and FBPase1 would take us towards gluconeogenesis. And these are going to be allosterically regulated by citrates, ATP, AMP, and ADP. So citrate, that's one of the components of the citric acid cycle. So basically, if we were to go through glycolysis, downstream of this, we would have citrate. So basically, if we're making enough energy, this is going to feed back and say, okay, PFK1, stop sending stuff down glycolysis, we've got plenty. But what if we don't have enough energy? We would still want to keep doing glycolysis. And so if we don't have a lot of ATP, if we have instead a lot of ADP or AMP, these are then going to go and stimulate PFK1 to take us towards glycolysis. If we have a lot of AMP, we don't have a lot of energy, and so we're not going to want to go and make glucose instead of breaking it down, so let's inhibit FBPase1. So the same molecule, AMP, can regulate both of these. Another regulator of PFK1 as well as FBPase1 is going to be this molecule called fructose 2,6-bisphosphate. It's another one of those allosteric regulators, and it sounds like it's something that's just like from your glycolysis pathway. Well, there we had fructose 1,6-bisphosphate, and here we have fructose 2,6-bisphosphate. So when you see bis, basically this is saying 2, it's got these 2 phosphate groups. If you see like diphosphates, like ADP, adenosine diphosphate, that would be the phosphate groups attached to one another and like in a row. But when we see bisphosphate, they're in 2 different places. And in glycolysis, we get this fructose 1,6-bisphosphate, and here we're talking about fructose 2,6-bisphosphate. Now, both of these are going to be made from fructose 6-phosphate, which was a product of the glycolysis. So this is kind of like a sidetrack from that glycolysis that's going to give us this molecule, F26BP. Now, F26BP is able to allosterically regulate both PFK1 and FBPase1, but in opposite directions. So when it binds to PFK1, it increases its affinity for fructose 6-phosphate. And when it binds to FBPase1, it reduces its affinity for its substrate. PFK1 activity in the presence of F26BP is going to be higher, but the FBPase1 activity in the presence of F26BP is going to be lower. So we said if we have F26BP, we're going to stimulate PFK1, and this is going and inhibit FBPase1. So this way we don't have this like fetal cycle. We're regulating them both at the same time, and we're doing so in a way that's going to favor glycolysis. If we don't have F26BP, well, now basically things are going to be the opposite. We're going to be favoring gluconeogenesis. And so we can control the levels of F26BP accordingly. And this is done through hormonal control. So going back to our idea of like insulin, and insulin's kind of opposite, glucagon. So we want gluconeogenesis in response to glucagon. If we want gluconeogenesis, we want to have low levels of F26BP. Whereas if we have insulin, lots of insulin, we're going to want high levels of F26BP. And so it turns out that basically what can happen is that the making of this F26BP is done by this really cool enzyme that's actually this 2 parts, or it's bifunctional. So it's got a phosphatase domain and a kinase domain in the same protein. It's really cool. So basically it has this kinase domain that's going to take fructose 6-phosphate, phosphorylate it to give you fructose 2,6-bisphosphate. And then it has a phosphatase domain, which is going to take fructose 2,6-bisphosphate and dephosphorylate it to give you fructose 6-phosphate. What determines whether it's acting as a kinase or whether it's acting as a phosphatase is its own phosphorylation state. When it's phosphorylated, basically, you're going to activate the phosphatase domain. This is going to then decrease the levels of F26BP, and this is going to inhibit glycolysis and stimulate gluconeogenesis. Alternatively, if it's not phosphorylated, well, now you're going to have the kinase domain active. You're going to make F26BP, which is going to stimulate glycolysis and inhibit gluconeogenesis. So if we phosphorylate this enzyme, we are basically going to be inhibiting glycolysis and stimulating gluconeogenesis. If we dephosphorylate this enzyme, we're going to stimulate glycolysis and inhibit gluconeogenesis. And so now we need to go back and think about when would we want to do what. Well, in response to glucagon, I signal that there's low blood sugar. we're going to want the liver to basically increase the amount of gluconeogenesis that's going on and stop doing glycolysis. So we would want there to be phosphorylation of this enzyme. Glucagon is going to act through a G-coupled protein-coupled receptor to make cyclic AMP. That cyclic AMP is going to activate the cyclic AMP-dependent protein kinase, which is then going to phosphorylate this protein, inactivate its kinase domain, activate its phosphatase domain, and basically then inhibit glycolysis and stimulate gluconeogenesis. To remove the phosphate, well, here it's going to respond to insulin, so basically the opposite of glucagon. The insulin is going to activate phosphoprotein phosphatase, which is going to remove this phosphate group to stimulate glycolysis and inhibit gluconeogenesis. So long, complicated, all this sorts of regulation, but is a great example of reciprocal regulation being used so that you don't have this like feudal cycle, making and breaking glucose at the same time. And finally, let's talk about this last step. In glycolysis, we take phosphoenolpyruvate and we take the phosphate group off of it and put it onto ADP to give us ATP. This is carried out by pyruvate kinase. In gluconeogenesis, this is the one that happens in those two steps. We have our pyruvate carboxylase step that's going to take us from pyruvate to oxaloacetate. And our PEP carboxykinase step is going to take us from oxaloacetate to phosphoenolpyruvate. So that glycolytic enzyme, pyruvate kinase, it's going to be allosterically inhibited by ATP, as well as in our liver, we'll see that it's actually going to be regulated by phosphorylation as well. So we have different versions of this pyruvate kinase. We have in our liver, we've got this L form, and then like in our muscles, we have this M form. Both of those are going to be regulated by ATP, but then also in our liver, we're going to get this phosphorylation regulation, which we'll get to in a second. Why does it make sense to be regulated by ATP? Well, basically, if you take pyruvate, you can go different directions. You can go and you can take it through the citric acid cycle, and you can generate energy. You can make ATP. Or you could go and you can take it to oxaloacetate. You can go to gluconeogenesis and make glucose. If you have a lot of ATP, basically the way you're going to get ATP is if you were to take it through to the citric acid cycle and then to oxidative phosphorylation. And if you have a lot of ATP building up that way, well, maybe it's time to go a different way. And so the ATP is going to negatively regulate this pyruvate kinase. In addition to be regulated by ATP, it's also regulated by other downstream things. So it can be regulated by acetyl-CoA. And not only can acetyl-CoA be made from the oxidative decarboxylation of pyruvate, it can also be made from the breakdown of lipids. It can be regulated by alanine, which can be made from pyruvate, by long-chain fatty acids, basically a lot of different signals that we have enough energy. And so let's go and let's do other things like make glucose. It also gets positively regulated, though. One of the things that actually regulates it is F16BP. So we just talked about how F26BP could be used to regulate things. Well, F16BP, that was actually one of the products of glycolysis. So basically, up here, we have this F16BP being made, and it's going to be activating this kinase at the very end of the pathway. Why does this make sense? Well, remember that this was our committed step. And so if we're saying, okay, we're pumping a bunch of stuff into this pathway, get ready for it. And so it's going to say, okay, get ready, pyruvate kinase, get active, because we've got a lot of stuff coming your way. And so that's why you get this kind of positive feedback from earlier on in the pathway, as well as the negative feedback from later on in the pathway. And then in our liver, things are going to be a little more complicated. We still have that same regulation, but now we're going to have an extra layer. Because in our liver, we can do gluconeogenesis. And so in our liver, basically, it's going to respond to glucagon. So it's going to respond to that hormone that says that we have low blood sugar. And our other cells, they're not going to have the glucagon receptors, but our liver is. And that glucagon receptor is going to act as one of those GPCRs. It's going to go through and, like we talked about before, give us the pathway where basically we can make cyclic AMP. That's going to activate our pKa, or protein kinase A, that's going to phosphorylate pyruvate kinase, the liver form. Now, this when it's phosphorylated, it's inactivated. So we see a lot of instances in which phosphorylation activates something. In this case, it deactivates something. So it deactivates pyruvate kinase. If you're deactivating pyruvate kinase, well, here this is going to prevent things from the pyruvate from going into glycolysis, and instead let it be used to go the other way and make glucose from it. And so that's how we can regulate the pyruvate kinase. How do we regulate the other things? So we have our pyruvate carboxylase and our PEP carboxy kinase. Pyruvate carboxylase, so that one that was taking pyruvate to oxaloacetate, it's going to be allosterically activated by acetyl-CoA. So remember, acetyl-CoA was going to be inhibiting the pyruvate kinase. And so it was inhibiting the upstream path of this from glycolysis. It's also actually going to be inhibiting the step from pyruvate to acetyl-CoA, so that things are kind of like, if you do get to pyruvate, don't go forward into glycolysis if you already have a bunch of acetyl-CoA. But if you have a bunch of acetyl-CoA, okay, now let's stimulate it going the other direction. Let's stimulate that pyruvate carboxylase to get us oxaloacetate. To go from oxaloacetate to our phosphoenolpyruvate, we need that PEP carboxy kinase, and it turns out that it's mostly going to be regulated transcriptionally. So our insulin, it didn't just activate that one kinase and stuff. It actually goes through pathways that are going to activate transcription factors. So it's going to activate proteins that are going to regulate the expression of genes. And it's going to regulate the of a bunch of different enzymes, including decreasing the expression of PEP carboxy kinase. So it's basically insulin is then able to turn off the things that you need for gluconeogenesis and turn on things that you need for glycolysis. And then we have other hormones, other transcriptional regulators that can stimulate making things for gluconeogenesis. So there's all sorts of transcriptional regulation that goes on there. Now let's go and identify various examples of these regulatory strategies at play in these pathways. Well, the one with the most complicated regulation is going to be this step, that's that committed step of glycolysis. This makes sense because we want to regulate tightly the entry into a pathway that is going to be basically, once we go in that pathway, the other alternatives are going to be a lot harder to go to. And so this is why we tightly regulate this phosphofructokinase and fructose 1,6-bisphosphatase 1. We can regulate this both by responding to the negative feedback to downstream products, so like responding to make our supply match our demand, and responding to our demands, increasing our supply if we need to. So basically, if we're running low on our products, make more. If we're running low on our reactants, but with a lot of products, well, then let's make less, and let's instead use these things for other pathways. We can have these same molecules, therefore, be negatively regulating one pathway and positively regulating another pathway. An example of reciprocal regulation, as well as integration between these different pathways. We also saw how we had examples of hormone-based regulation in this step, where we had glucagon and insulin be controlling the making of a metabolic modulator, this F26BP, this allosteric modulator. We could have these hormones basically acting through phosphorylation relays, affecting these multifunctional enzymes that have these complicated effects that make this allosteric product, which then has opposing effects on opposing enzymes. Because we want to reciprocally regulate these enzymes so that we don't have a fetal cycle where basically we're making and breaking at the same time, and all it is doing is generating heat, not any beneficial energy or anything like that. So all of those were examples of regulatory strategies. We also talked about how we can have compartmentalization and specialization, where certain cells would have receptors, so that our liver had receptors for glucagon, and it had those enzymes that were specialized for responding to the metabolic needs and the functions of the liver its requirements to supply glucose to the rest of the body. We saw how it had basically this different hexokinase, this glucokinase with a higher Km, so that it's going to still allow glucose to get out and not hog it all. And it's able to respond to changes in glucose concentrations. In our other tissues, we had this lower Km, a tighter affinity, so that although those cells were seeing less glucose, they were able to trap it more efficiently. We also saw how we can have other pathways relay this kind of like similar information, so that even if we don't have energy being formed from glycolysis, if we have a lot of ATP building up from other pathways, as well as things like acetyl-CoA building up from other pathways, that can also be a signal that we have plenty of energy, and therefore we can go a different pathway. So integration of signals from both inside of the cell, from outside of the cell, through hormones, different specialization in different tissues, allosteric regulation to the max. All of these things are on display in these pathways. And then all of these things are also in display in a lot of other pathways that we're not going to have time to get into. But hopefully you can apply the same logic and those pathways will seem more, I guess, logical.